My talk today will be focused on using AI to accelerate drug discovery for COVID and also our ongoing COVID-19 global pandemic efforts. Um, so a little bit about Insilico, just one quick slide. So we are 120 people worldwide, about 80 AI scientists. We're global. I'm dialing in from Hong Kong today, where our headquarters are, but we have multiple global offices worldwide. We hire primarily through competitions, national competitions, and then train people in chemistry and biology. So we basically take AI guys in and then train them in chemistry and biology. And we raised quite a bit of money from very sophisticated investors. We specialize in novel target discovery and novel molecule generation. Primarily, we focus on generative adversarial networks and deep reinforcement learning. I'll briefly cover this technology, and we try to originate our own IP. So in a nutshell, we specialize in target discovery. So we try to identify the mechanism behind a disease and narrow it down to a specific set of targets where we can intervene. Then we generate novel molecules that do not exist in known chemical space, and try to target those targets with those new molecules, validate and take some of those molecules into preclinical work. We also focus on clinical trials outcomes prediction. So one of the main technologies that we specialize in is generative adversarial networks. This is not the seminal paper, but one of the first papers that visualized this technology in a very, very sophisticated way. I highly recommend reading that one. It's ICML 2016 by Reed. And it shows you how two deep neural networks, one is a generator and another discriminator, are competing with each other to produce meaningful objects. So you can see here that the generator taking in input parameters like this flower has small round violet petals with dark purple center. So textual input generates meaningful noise, and the discriminator, which is trained on also millions of images, is trying to recognize whether the output of the generator is true or false, comparing it to ground truth. So in this kind of contest, that's why it's called adversarial, these networks learn to produce this kind of output. And again, this is 2016, very early days. Now those systems are perfect. But here, this paper showed that you could basically give it some text. This small bird has a pink breasting crown and black primaries and secondaries. And you can see that the direction of what you are trying to achieve is there in this picture with human intelligence. And in 2017, those networks became so good, GANs became so good that you would not be able to recognize a GAN-generated face from the real person. And nowadays, this technology is used in video games and movies pretty much ubiquitously. And we use this technology in biology quite a bit. Again, it got so good that it's much better than legacy realistic videos. And you actually can see me here dancing Bhangra at the Indian Heritage Day in Kingston, Ontario. I was a student at Queen's, so really love those dances and music. Many of the concepts in deep learning date back to the 1940s, 1950s, and then late 90s and early 2000s. GANs are a pretty new concept, originated in 2014 from the work of Ian Goodfellow at that time at the University of Montreal and then at Google. So we started using this technology for generation of novel chemical matter that does not exist in the known chemical databases with specific properties. So, for example, I want to have a molecule that inhibits protein A with at concentration X with bioavailability Y. So with many, many, many different parameters, and this networks produce those chemicals, those molecules. And at Insilico, we have two strategies for attacking COVID-19. So one is generative chemistry, basically generating molecules that go after very specific targets in COVID-19. So those are the targets encoded by the SARS-CoV-2 genome. And we also have a tool called COVIDomic, and it's an efficient tool for stratification of patients that come into the hospital into the patients who are going to have severe symptoms and those that are likely to be very mild. And this system is also very good. It's a predictive model. So I'll cover the section first on generative chemistry and antiviral drug discovery and our current status. So if you look at the various phases of disease progression, we can intervene by preventing the disease, preventing it from progressing from exposure and high risk of infection. We can address early symptoms, acute phase, recovery, rehabilitation. So we are going after many of those areas. But one of the areas which is most likely to work in the long run are the viral proteins that are encoded by the viral genome. One of those proteins, again presented here, is C3-like main protease, which is responsible for viral replication. Of course, there are many other key viral proteins that are not present in our genome, but are present in a virus. So if you inhibit them in a very targeted way, you are likely to either slow down or kill the virus. And we are going after this C3-like protease, which is responsible for replication. There are multiple published crystals for this protein. And what we did in the past, in 2018, we did a few case studies, and I highly recommend looking at this paper. It actually got quite a bit of coverage, probably more than it deserves, because we did many other really cool things. But in this paper, we show how, given a protein target, we utilize a system called generative tensorial reinforcement learning system that very, very quickly goes from the presentation of a molecular target to a molecule that can be synthesized and tested that would hit this target. And we used this technology for COVID. So in April, May timeframe, we took just 4 days to generate about 100 molecules for some of those COVID-19 specific proteins, C3-like protease. We put it on archive and enabled some of the human medicinal chemists to go in VR with a company called Nanome and comment on those molecules. So many of them liked it, so they massaged them. And then we published those molecules. And those molecules, again, they do not exist in the known chemical space. So you can see that there are no records that are similar, at least by 40%. So we are trying to go after molecules that are not present in the known molecular space. They are completely from scratch generated and invented by those GANs. And it's modern chemistry. When medicinal chemists look at them, they actually like them, so they not vomit when they see some structures that they know that they're toxic or non-synthesizable or just very old. And some of our work was recognized by Intel, and Intel has an amazing team that is part of this COVID response and pandemic readiness initiative. They have really top brass people from all walks of life who are monitoring the situation and contributing to the effort, but they're also contributing to the projects that are likely to be useful for other pandemics or for just healthcare in general. And we started collaborating with some of those people, and they supported our synthesis initiative, where we actually discovered. So here, when you see the ID in this column, you see the IDs of the molecules that we actually synthesized. So they were created by AI. We synthesized them and tested them in a petri dish in vitro against the viral SARS-CoV-2 protein, C3-like protease. And this inhibition, so when it shows green, it's very good. It's very good inhibition. And then we went and tested it in cells and also in mice. And you can see that for some of those molecules, they could be really good drugs. And again, those are novel. Unfortunately, again, AI is kind of a Ferrari that gets you from 0 to 100 very quickly in seconds. But after that, you have to move with the speed of traffic. So you have to manufacture those molecules. You have to put them into mice. You have to then put them into humans. So it's a long process that you cannot really kind of avoid. And but the fact that we cut down, maybe a few years of discovery time, it's pretty impressive. And this slide shows you the kind of process that we've taken. It took us only 4 days to generate those 49 compounds that were later synthesized and tested, and most of them were found effective against this protein target that we're going after. So this is a pretty impressive feat. We hope to be able to publish it soon. And again, there are some compounds that are known to hit the C3-like protease, but they are reasonably old, and many of them have a lot of problems. So they do not really, they make really good publication materials for journals, but they do not have a lot of drug-like properties that we need in order to put this protein inhibitor on the market as a drug. And we can see that our AI-generated molecules do possess many of those properties. Again, it's too early to say. It would be enough for a good paper, but we want to take it further into late preclinical, and then hopefully put it on the humans. So, and then we have another. I'm switching gears into multi-omics analysis that we have done also with the help of Intel team. And we are deeply thankful to them for allowing us to go into this direction. And we built a tool called COVIDomic. It's an AI for patient stratification to predict severity and mortality. And for this, we use metatranscriptome analysis from the patient, basically microbial data from nasal swabs and COVID-19 genomic data. We also use patient metadata, and we use blood tests to predict severity, survival, disease course analysis, and also look at many different risk factors that are associated with COVID-19 patient journey. For that, for a pilot, we used 3 different data types. So blood biochemistry data types, SARS-CoV-2 viral genomes, metagenomic data. Actually, at Insilico, we're pretty much like an MMA fighter for all data types. I do not discriminate any data types. I really like to have all data types integrated. Sometimes we even look at facial pictures that contain more information than a genome. But for this pilot, we use those 3. And since there are usually not enough data available in the public domain or actually even in collaborations, we started using those deepfake technologies that I've described before, GANs, generative models, to take small data sets, but high quality data sets of patient metadata, microbial data, COVID-19 genome data. And then we use those GANs to generate a lot of those patient samples with different conditions. So here is an example. You take just 65 initial samples of very basic blood tests that are annotated by severity and other features. And you can see the distribution of the 65 original ones. And then you use a generative model and generate 10,000 samples. And those 10,000 samples can be used for many other applications like other model training or working on a population level, combining with different data sets where you have more data available with specific features. And we are doing just exactly that. So in blood biochemistry data, when you are using very simple parameters, that's something that any doctor can obtain from a patient, so any general practitioner, we show that the generated data is so good that it's almost indistinguishable from the real data. The generated samples follow the very expected pattern of Fisher behavior correlation. And you can see that each cell is a correlation value has a correlation value between -1 and 1, and we see that oversampling doesn't create any uncommon correlations. We have very strongly correlated features like bilirubin here. Only one feature, one of the features would be useful for final model training. This is very high quality synthetic data. We can also see that many features from the blood test correlate very well with severity. So there is a clear correlation between the features that are coming from the blood tests and severity of the symptoms. And one really cool idea that I came up with in 2015 is that when you don't have a lot of data about the patient, when you don't have a lot of patients with a specific disease, all of them have age. So usually age is the most ubiquitous feature in any data set. So if your data looks like Swiss cheese, age is usually there. So what you can do is you can train deep neural networks on various data types to predict age first, to do many other things, to basically derive pathways, targets, causal graphs, synthetic data, just also for proof of concept. But then you can retrain this network on a much smaller data set of patients that are annotated with the health status, with disease. And then the deep neural network, you can do transfer learning from age to disease. And that was a pretty big discovery. So I published a paper in 2016 that became quite popular. And we started doing a lot of work in this area. Also basically linking, so all of us are going to die, regardless of how you look at it. And in the process, we're going to decline, get old, get diseases, etc. So we want deep neural networks to capture this biology on many data types and figure out how we can avoid the various risks. And we built a bunch of aging clocks for this purpose based on microbiome, transcriptome, blood tests, even pictures. You can use this system right now at young.ai website, or there is an app, young.ai. You can play around with it. We just launched. And you can really clearly see that an individual feature like albumin or cholesterol does not significantly predict age. It does not have a lot of predictive power. But if you combine them, if you take, let's say, 41 normalized markers and start predicting age, you can get with deep neural networks with one neuron on the output, just the age, and you ensemble those models. You can get to pretty reasonable accuracy, mean absolute error of 5.5 years. I sometimes cannot predict people with this accuracy, but the deep neural network based on blood can do it very well. We started noticing that people who are predicted to be older than their chronological age, they usually have some problems. They usually have some comorbidities. People who are predicted to be younger, they're actually quite fit. And those predicted ages are very correlated with mortality, with hazard. So if you are predicted to be 5 years or older than your chronological age, your hazard ratio, your all kinds of risks leading to death are much higher. So in some cases, like 5 times higher. And if you are 5 years or more younger than your chronological age, the probability of dying of any cause is lower. So that was a pretty cool paper as well. So we used this same concept to predict severity and mortality in COVID patients. And it works very, very well. You can see that people who are predicted to be older, they are very, very nicely clustering into the severe clusters if you look at those t-SNE plots. And I'm out of time, so I'll skip a few slides right now. But actually, you can base decision trees based on combinations of those features and combinations of these predicted ages. And then you can start combining many of those data types together and training together or separately on a variety of data types and combining those scores to get very, very nice prediction of severity. So you can see that if I take an ensemble of 2 models just trained on viral genomic data and blood biochemistry data, I can predict severity with 94%. So 94 F-score accuracy. And now I can also use very, very simple blood tests. So we do some feature extraction and look at the importance of those features. So we can predict severity with very, very high accuracy. And in the future, in the very near future, we're going to use much simpler data types, even hopefully pictures, to be able to predict severity. And now we are building this COVIDomic platform, which is running on a very large collection of data. A lot of this data is synthetic. And actually, one of the ways to anonymize data to ensure that you can go away from individual data and you don't need to care about privacy as much is to synthesize the data of the same patient just 2 years later. And maybe several other features can be different. But the core features that relate to the disease stay the same. So we have a very large collection of this data. And we are building this platform to enable people to predict to predict severity, mortality, and many other features. But also we open the API for other people to use this system to be able to publish really nice papers, but also to hopefully save lives. And this system is going to be open. So part of it is already open. So thank you very much.